Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will talk about negative staining. Negative staining technique is used in microbiology for staining the background. Staining the background. In this method, we use acidic stains that contain negative charge on them. So, we're using acidic stains that contain negative charge on them. So due to repulsion between bacteria having a negative charge and a stain containing also negative charge, so there will be repulsion between charges and bacteria cannot be stained. Resulting bacteria will be colorless, whereas all the background of bacteria will be stained. So this staining process resulting in this staining of the background. Examples of acidic stains include nigrosine and Indian ink. In this method, we don't apply any kind of heat, so it's a complete heatless method. In this method, we are not using heat because heat usually damages the bacterial structure and causes the bacteria to shrink. So in this method, we are not using any kind of heat, so we can determine the more accurate size of bacteria. Also, after the staining method, all the bacteria will be colorless, so we can identify whether a bacteria is a round shape, that means that is a cocci, or a rod shape, that is a bacilli. Also, this method can be used for determining whether a bacteria contain capsule or not. So all the bacteria that containing a capsule will appear as a will show a hollow, right? After this method, whereas all the background will be stained except, for example, if this is a bacteria and this is the capsule of bacteria, all the background will be stained, but around this capsule area there will be a hollow scene, right? A complete unstained area. Also, negative staining method can be used for detecting fungi known as cryptococcal neoforman. This fungi contain capsule. This is a capsulated fungi. So after staining this method, these cryptococcal neoformans will be round shape, have a round shape structure and will appear as colorless against a dark background. Negative staining method can also be used for observing spirochetes. Spirochetes are bacteria that are extremely thin and can't be observed using normal staining technique and light microscopy. So in order to observe them, we can use negative staining procedure, and spirochetes will appear as colorless entity against a dark background, so we can observe them under the microscope. So now we'll talk about the procedure that is very easy for negative staining. Now we'll talk about the procedure for performing negative staining. It can be done by two ways. First, it can be done negative staining using a spreader method. First, we'll talk about this method. In this method, we will take a clean glass slide, and near the edge of the slide, we will put a drop of Indian ink or nigrosine or acidic dyes. Then we'll pick our colony with the help of loop, wire loop, and then we'll mix with this dye, right? Then we will take another slide. This slide will be called as spreader slide. For example, this was our first slide. We have put a drop of Indian ink and we have mixed it with the suspected microbe. That can be bacteria or fungus. For example, fungus include cryptococcal neoformans. And then we'll take a spreader slide. For example, this is our spreader slide. We'll just uh, put on the surface of first slide and touch this solution, right? Solution of dye and a suspected microbe. Then after touching this solution will be spread along the surface of the slide. Then we will just drag it, and after dragging it, we will make a smear that will be very thick at this end and thin will be another end, right? We will let this smear air dry, then we will examine it under the microscope. Between a thin and a thick smear, there will be a zone. uh, that zone is not too thick or not too thin to be observed under microscope. Now, talking about the second method for performing negative staining using wire loop or inoculating needle. In this method, we'll take a clean glass slide. Near the center of slide, we'll put a drop of Indian ink or ribosine. Then we'll pick our suspected microbe and mix it with the dye. For example, this was a dye using a wire loop. Wire loop look like this, right? Using this wire loop, we'll pick up a suspected microbe and mix well with this dye, right? After mixing, we will spread this solution over the surface of slide and make a smear, right? So this will be our smear. Then this smear, we will let this smear air dry so water will evaporate, right? And we can see after air drying the slide, we can see it under the microscope. This was the procedure for performing Indian ink using inoculating needle or wire loop.